potential. The symbol for that is the Greek letter psi, and this is a pre-lab for AP Bio number 4. Water potential describes the tendency of the water to move from one place to another. Water moves from high water potential area to low potential area. The water potential depends on pressure, as well as depends on the solute concentration in the solution. Or we can write it down, water potential equals the pressure potential and the solute potential's sum. So P means pressure potential and CS means solute potential. Solute potential, the solute's concentration in the water. So here is going to be a cell. What pressure potential means, this is a cell, and the cell wall, in this case, or the cell membrane too, can exert pressure on the solution inside of the cell. So the pressure on the solution. The other one is the solute concentration in the water. It is given that the water's potential, when there's no solute in it, it's 0 at normal atmospheric temperature. So when we add solute to the water, its potential is going to be less than 0, so makes the water potential low. The solute potential is always negative. We will see that later. The unit for the water potential is bar, or the atmosphere. ATM. Okay, anyway, calculations. Try to figure out the solute's potential. The solute's potential is a big equation, but don't worry, you don't have to memorize it. I stands for ionization constant. The C stands for molar concentration. R is the pressure constant. And the temperature is going to be, the T is the temperature is going to be measured in Kelvin. Now, here is the sheet that you are going to get at the AP exam. And you do not have to memorize those because they are on the sheet, but you have to know how to apply the equation. So, problem 1. We have a 2 molar glucose solution at 25 Celsius. Let's find out the solute's potential of that solution. So, you just plug into the equation very fast. And I made a mistake at the first calculating out, so we will wait a little bit. It's not going to be 48.22, but it's going to be minus 49.53 bars. Second problem. Now, 1 molar sodium chloride solution. At this point, the ionization number is going to be 2 because the sodium chloride dissociates it into its ion, the sodium and a chloride ion, and that's why it's going to be 2. The glucose in the previous one did not change. It was 1 because 1 glucose will not turn into 2 ions because it's covalent bond. But anyway, so it will be 1 glucose still. So the solute potential is going to be minus 4870 bars. The problem, what you have to use later on in the lab. The question is, which way would the water go? Here is a solution, the sodium chloride solution, and this is 0.2 molar at 25 Celsius. Find out the water's potential. In order to find out where the water would go, we have to figure out the water's potential. How are we going to figure out the water potential? We need the solute's potential first, and we are going to plug in minus 2 again because sodium ion and chloride ion. The molar mass the molar over the liter, the molar concentration, the constant, the temperature, and it will be negative 9.9 bars. The pressure potential will be 0 at this time because it's open container. So the water's potential is minus 9.90 bar. If we place a cell or something, in your case it will be a dialysis membrane with solution. If we have a cell or something like that inside of that solution, that it has solute potential as well as pressure potential, we figure out the water potential is minus 1.6. You figure out which one is higher and which one is lower. Higher is in the cell, so the water is going to go out from the cell into the surrounding solution. That's what we are going to apply with the lab.